Welcome back to the Explainer. So, have you ever popped a headache pill and wondered why on earth it takes, 30 solid minutes to kick in, while other drugs, like certain anesthetics, can literally knock you out in milliseconds? Today, we're diving deep into the microscopic world of pharmacology. We're going to track the absolute incredible speed at which different drugs interact with our cellular locks.
All right, let's just make sure we're on the exact same page before we get too deep. What is a receptor really? Think of it as a highly specific lock, just sitting there waiting for its perfect biochemical key. Now, whether that key is an endogenous hormone your body naturally makes or an exogenous drug cooked up in a lab, the receptor, which is usually a protein, is the macromolecule that actually recognizes it. And the moment that perfect key slides into the ligand binding domain, boom, it triggers the effector domain. You can think of the effector domain like a complex set of gears turning inside the cell, ultimately creating a biological response we can actually measure. Here is our game plan for today. First, meet the cellular locks. Then, type 1, the lightning fast. Next, type 2, the second messengers. After that, type 3 and 4, the slow burn. And we'll wrap up with number 5, when cells fight back. Let's jump right into section 1, meet the cellular locks.
So, looking at these four main types of receptors, you'll immediately notice this really natural, almost beautiful deceleration in the response speeds. We start with the lightning-fast type 1 receptors, dealing in pure milliseconds. Then we drop down to type 2, operating in seconds to minutes. Things drastically slow down with type 3, taking anywhere from minutes to hours, until we finally hit the marathon runners, type 4 receptors, which can literally take hours or even days to show their full effects. And if you're wondering why there's such a massive difference in timing, well, it all comes down to the actual machinery attached to the lock itself.
Which brings us to section 2, type 1, the lightning fast. So we're looking at the absolute fastest responders in the human body here. Notice how the receptor is oligomeric? That just means it has 4 to 5 subunits clustered directly around a central ion channel pore. And honestly, this is the entire secret to their blistering speed. The receptor and the ion channel are literally part of the exact same protein complex. There is absolutely no middleman here. Shuttling ions, like sodium, potassium, or chloride, happens instantly. And this direct ion flux is exactly why drugs like ketamine or diazepam can cause a massive systemic response in mere milliseconds. Let me walk you through exactly how this rapid-fire process plays out. Step 1, the ligand binds to the outside of the cell. Step 2, the protein immediately changes shape. Step 3, that central pore pops open instantly, letting ions just flood in or out along their electrochemical gradient. This immediately shifts the membrane potential, which gives us that millisecond cellular response. In step 5, it's like the second the key turns, the door gets kicked wide open. And then, just as quickly in step 6, the ligand detaches and the channel snaps right back shut, totally ready for the next signal. All right, moving on to section 3, type 2, the second messengers. We're slowing down just a bit now to enter what I like to call the middle management of the cellular factory. These G-protein-coupled receptors, or GPCRs for short, deal specifically with hydrophilic drugs that can't just easily slip into the cell on their own. Because they rely on a middleman, a G-protein, to send second messengers like CAMP or IP3 deep into the cell, the whole response takes seconds to minutes. Basically, they have to pass the memo along a chain of command. The structure of a GPCR is honestly fascinating. It's this single, continuous polypeptide chain threading its way in and out of the cell membrane exactly 7 times. That's why you'll often hear them called 7TM or serpentine receptors. So it just sits there, waiting for a drug to bind on the outside so it can shift its shape. Once activated, it wakes up that waiting heterotrimeric G protein on the inside, splitting its alpha, beta, and gamma subunits apart to go out and execute the cell's orders. Now, when that internal G protein is a GS, or stimulatory type, it essentially hits the gas pedal. It activates an effector enzyme called adenylyl cyclase, which then spikes the production of our second messenger, CAMP. The actual biological effect? Well, it activates protein kinases that do things like cause bronchodilation or crank up your heart rate. This is exactly the pathway that lights up when adrenaline, or a drug like dobutamine, hits the beta-1 receptors in your heart. Conversely, the G pathway acts as the brakes. It actively inhibits adenylyl cyclase, bringing CAMP levels way down. This lowers your heart rate, causes vasodilation, and inhibits neurons. If you've ever wondered how opioid drugs like morphine actually kill pain, well, this is it. They bind to mu receptors, which triggers this exact G pathway to shut down those pain signals.
Finally, we've got the GQ pathway, which takes a completely different road. Receptors like alpha-1 or histamine H1 trigger this GQ protein to activate something called phospholipase C. This creates totally different second messengers, specifically IP3 and DAG, which cause a massive spike in intracellular calcium. And that calcium spike, that's what triggers heavy-duty actions like smooth muscle contraction and glandular secretion.
Okay, on to section 4, type 3 and 4, the slow burn. Now we're really drastically slowing down our biological clock to look at the long-term systemic changes. Type 3 kinase-linked receptors take anywhere from minutes to hours to fully respond. The receptor itself actually contains an intracellular kinase domain, most commonly tyrosine kinase. Receptors like EGFR or ANP live in this category, which means drugs like insulin or targeted cancer therapies like trastuzumab and gefitinib are binding right here. And the reason it takes so much longer is that they have to kick off this incredibly complex multi-step phosphotyrosine cascade just to fundamentally change how the cell metabolizes or grows.
If you trace these steps, really pay attention to step 2. This is wildly different from everything we've seen so far today. For type 3, two completely separate receptor halves, which we call monomers, must physically find each other in the membrane and bind together to form a dimer before any work can even begin. Only then can they cross-phosphorylate each other to create docking sites. This finally triggers the downstream cascades that eventually lead to sweeping biological changes in gene expression. And that whole setup process is exactly why this takes minutes to hours.
Arriving at our absolute slowest timeline, type 4 nuclear receptors aren't even on the cell's surface. They're actually hidden deep inside the cytoplasm or the nucleus. Because of this, their ligands, which are highly lipid-soluble, like glucocorticoids, estrogen, or thyroid hormones, have to bypass the cell membrane entirely. They literally float inside, find a receptor, enter the nucleus, and directly rewrite the cellular DNA through gene transcription. This kind of intricate genetic reprogramming is exactly why it takes hours or even days to actually see a biological effect. And that brings us to our final section, number 5, when cells fight back.
So what happens when we just bombard these delicate cellular locks with the exact same keys, day after day, year after year? Well, our cells adapt. They constantly adjust their sensitivity. Take downregulation, for example. If you overstimulate a cell with an agonist drug, like someone overusing a salbutamol inhaler for asthma, the cell defends itself by literally pulling receptors inside. It internalizes them, which causes drug tolerance. But on the flip side, upregulation happens when you chronically block a receptor with an antagonist. The cell essentially panics and builds extra locks. So if you were to suddenly stop taking a drug like a beta blocker, all those new, highly sensitive locks are suddenly exposed, which can lead to a really dangerous rebound effect, like sudden tachycardia. And it gets wilder because this desensitization actually comes in 2 flavors. Homologous desensitization is highly specific. It uses GRK enzymes to target and silence only the specifically occupied receptors. So the cell only becomes insensitive to that one specific drug. But heterologous desensitization, that uses pKa or pKc to phosphorylate even the empty, totally unoccupied receptors. This creates this massive, sweeping cross-tolerance to multiple different drugs at once, which, by the way, is exactly why opioid tolerance can be an absolute nightmare to manage medically. So I want to leave you with a thought, really pondering the future of pharmacology. If we can map every single adaptation these microscopic locks make, from the millisecond ion flashes we talked about, all the way to the days-long DNA rewrites, can science eventually forge a master key? You know, a drug that perfectly treats the body without ever triggering the cell to fight back and build tolerance? It is absolutely the ultimate quest in medical science right now. Anyway, thank you so much for joining me on this explainer, and remember to keep questioning how the amazing world inside you actually works.